Welcome to week 5 of viruses, where I'll be talking about oncolytic viral therapy. So we're just going to talk about a couple of background points before moving into the actual primary study. And then for the outline, rather than discussing, I guess, a discrete agenda, we're going to cover a couple of the questions that hopefully by the end of today's talk, you'll have the capacity to answer somewhat well. So number one is, why are solid tumors difficult to treat with immunotherapy? Right. So in a previous week, we discussed immunotherapy and how it seeks to augment the body's immune response to tumors and cancers in general. But now we're going to talk a little bit about the application of these immunotherapies and why they're less efficacious in some settings than others. Secondly, we're going to talk about what oncolytic viruses are and about what differentiates them from other approaches in the immunotherapeutic arsenal. So as you can probably gather by the name, oncolytic viruses are viruses that are engineered to lyse cancers or cancer cells selectively. And lastly, can we administer oncolytic viruses as combined immunotherapies? And if so, how does anti-tumor immunity change as a result? So move this window. I don't know how to make this go away. Okay, there we go. So broadly speaking, tumors provoke adaptive immune responses, right? We normally don't think about our immune system responding to cancer cells, but in fact they do. And I'll just walk us through the process of how that happens with this graphic on the left. So we see initially tumor cells dying as a result of chemotherapy, but really this can be extended to any situation in which a tumor cell might die. And when it dies, the cell membrane permeabilizes and it releases several danger-associated molecules, such as HNGB1 or extracellular ATP. And these sort of alert nearby sensor immune cells that are sort of surveilling for these events or these molecules in circulation or tissue. And so in particular, you have your immature DCs that can recognize these DAMPs and become stimulated to mature, in addition to taking up any locally secreted tumor-associated antigens and process them intracellularly to present them on their MHC proteins on their surface. And so once those dendritic cells are matured and are presenting that antigen, they migrate to your secondary immune organs, typically your tumor-draining lymph node, where they'll then interact with naive T cells and begin to activate them. And then skipping a bunch of steps, the activated T cells will migrate into the tumor and directly lyse these tumors with perforins, granzymes, in addition to secreting interferon gamma that can stimulate macrophage responses. But what's important to note here is that the tumor or the T cells that are activated and killing the tumor cells actually infiltrate into the tumor. So they're not just sitting at the periphery, but they actually squeeze their way into the tumor and attempt to reach as deep into the tumor as they can in order to maximize their cytotoxicity. And this degree of infiltration is something that's highly correlated with a response to immunotherapy, both as a prognostic marker and a post-treatment marker of efficacy. However, we'll soon learn, one, this is not always the case because the immune system is complicated, but also that not all tumors are created equal in terms of their capacity to allow for immune infiltration. Specifically, there are 3 buckets really that the cancer immune phenotype can fall into. Shown on the right, where we have immune-enriched tumors, where it generally follows a schematic where the tumors have high amounts of antigen, they're highly immunogenic, and they support deep infiltration of cytotoxic lymphocytes. Then you have your immune-privileged, which are sort of your intermediate phenotype, where they're slightly less antigenic and they have slightly reduced or perhaps vastly reduced number of immune cell infiltrates. And then lastly, you have your immune deserts, which are very poorly antigenic, and really have almost no immune cell infiltration. And so the question is, it sort of makes sense, right? That immunotherapies would work better in the immune-enriched tumors because there are more immune cells that can enter and exert their effector behavior. And then that immunotherapies would perform more poorly in immune desert tumors. And so the question in the field has really become, how do we convert immune desert tumors into immune-enriched tumors such that immunotherapies are more likely to succeed in these otherwise hostile environments? And again, we're glossing over a lot of the other factors playing into why these environments are hostile other than just antigen density. But that's for another day. Or actually, it's for this slide. The tumor so part of why solid tumors are so hostile to immune cell infiltration is their tumor microenvironment. And so shown here on the graphic, specifically in this bottom, is a list of several mechanisms by which solid tumors prohibit immune cell infiltration. So there's on a molecular level, there can be antigen escape, where they simply lose the antigen that the T cells would otherwise be targeting. They are also highly dense, and so their centers become very poorly perfused and hypoxic as a result, which causes anaerobic respiration, which leads to acidosis, and high levels of lactic acid, which promote T cell exhaustion and anergy and loss of effector ability. And in addition to that, and perhaps what's most crucial for today's talk is that tumors promote angiogenesis by secreting androgens, myriad androgens, which promote the development of vasculature into the tumor. But it's extremely important to understand that this vasculature is aberrant and highly deranged. And so while it does perfuse the tumor enough to sustain its growth, because they're highly deranged and extremely leaky, they are not conducive to T cells trafficking through them and into the tumor core. And in fact, it actually inhibits immune cell trafficking to the tumor. And there's my build. So now that we understand tumors and their microenvironment, now we need to talk about the other critical component of today's talk, which is the oncolytic viruses themselves. So oncolytic viruses can be understood as depicted in this graphic on the left, where they're viruses engineered to selectively target cancer cells and lyse them. The process of which releases PAMs and DAMPs, which can then go on to activate or recruit additional cytotoxic cells like your NK cells, your CD8 cells, in addition to primary dendritic cells. And the other thing worth noting is that they're highly efficient and they are self-propagating because they're viruses. And so you might think of these as fighting fire with fire, only your fire is a lot bigger and it grows a lot faster than the cancer cells. And typically these are administered intratumorally, not so much systemically. The reason being, if you administer them systemically, they're subject to clearance by the innate immune system, or just because in addition to lower levels of or poor pharmacokinetic properties, because they have to circulate the entire body, and that prolonged trafficking time makes them susceptible to clearance. And what makes them selective for cancer specifically is that they take advantage of the cancer cells' impaired antiviral machinery. So you can think of cancer cells as having gone all into the proliferation and survival metabolic pathways, whereas their ability to produce antiviral proteins and signals like interferons is severely impaired. And so what happens is these viruses, while they can infect your body's normal cells, your body's normal cells also have the ability to produce interferons, which can signal neighboring cells to produce molecules that will interrupt the viral life cycle should they be infected themselves. And so because the cancers have difficulty doing the same thing, they're much more susceptible to viral lysis and infection. And lastly, this really isn't relevant to today's talk, but it's very cool, so I wanted to bring it up. So there are DNA and RNA viruses, each with their own advantages. With RNA viruses, because they're much smaller, they're more able to penetrate the blood-brain barrier, and there are several groups using RNA viruses to clear glioblastomas, which are very aggressive death sentence, essentially. Whereas with DNA viruses, they actually have the ability to introduce transgenes into the cells they infect and cause the infected cells to produce pretty much anything. And so this is an exciting area of research, in my opinion, that is just starting to burgeon. And then lastly, wanted to point out that there has been one oncolytic virus approved for metastatic melanoma, and there are several clinical trials for its combination with other immunotherapies and chemotherapy. So it is an established mechanism, however, still a very young field. So continuing this, I wanted to point out that it has a high potential in combination with other immunotherapies because they release the PAMs and DAMPs. It's sort of acting to enhance the immune response in a way different, in a separate pathway from other immunotherapies. And so by enhancing immunotherapies or enhancing the immune cells through two separate mechanisms that don't necessarily overlap, you might stimulate a stronger effector response. And this is an incredibly crucial point, this third point here, that I really want to highlight for today's talk, that the majority of oncolytic virus-induced tumor debulking or loss of mass is immune-mediated, not virus-mediated. So what this means is that, yes, the virus kills cancer cells, but the majority of cancer cells that can be attributed to oncolytic viral therapy are actually killed by the immune system. And there's a ton of studies that support this. And it's interesting that it sort of has this two-pronged approach where it can engage your own immune system and have it kill the cancer cell in its place. And it really lends credence to the combination strategy, in my opinion. However, in addition to, or perhaps similarly to how immune cells are limited by poor vasculature, oncolytic viruses, because they are circulated and small, they are also similarly limited by tumor vasculature. And so poorly perfused tumors will have fewer oncolytic viruses, particularly if they're systemically administered. So this brings us to today's paper, which I will try to talk through as clearly as possible. I think it's from 2018 or 2019, where the authors studied Newcastle disease virus as an oncolytic virus and combined it with an anti-angiogenic factor to see if they could produce or generate a stronger immune response and subsequent tumor clearance. And the model they used were mice with metastatic ovarian cancer. But before they did their in vivo experiments, we need to talk about one of their introductory in vitro experiments, where they seeded 3 different cell lines into 12-well plates. So they have the ID8 cells, which are your ovarian cancer cells that are from mice, your CAOV3 cells, which are the human equivalent, and then your nose cells, which are actually normal human ovarian surface epithelial cells. So mouse cancer, human cancer, regular ovarian cells. And then they will infect with the Newcastle disease virus at a multiplicity of infection of up to 10. And so what this MOI represents is a ratio of virions to cells. And so this means there are 10 times more virions than there were cells. And then they waited 48 hours and counted the, or they didn't count them, they assayed the medical activity of the remaining cells as a proxy for cell survival. And what they showed here is that the oncolytic virus was highly selective for cancer cells, So in both the mouse and human ovarian cancer cell lines, there was a dose-dependent decrease in tumor metabolic activity, or not tumor, cell metabolic activity, whereas with the normal epithelial cells, there was a much gentler slope as dose increased. And so this suggests that there's a vastly different response between cancer cells and normal epithelial cells in the ovary. Now we move on to what is the bulk of their paper, which is their in vivo study, where they injected mice with the ID8 ovarian cancer cells. So these are the mouse-based ovarian cancer cells. Just let them grow for day 60 and presumably metastasize. And then they split the mice into 4 different treatment groups. So one was untreated. One, they administered 3-TSR, which is a recombinant thrombospondin. That's an anti-angiogenic factor. And they administered 4 mg per kg intraperitoneally every day from day 60 until the end of the experiment. In group 3, they had 1 dose of their oncolytic virus on day 74, which was also engineered to have a transgene for GFP. And then in group 4, they just combined the 2 with the same time points. And so they euthanized the mice 30 days after the initiation of treatment, so day 90. And they collected the primary tumor, and they also assessed the peritoneal metastases. So on the next slide, I'll walk through each of these panels. Number 1 is that relative to the other treatment groups, the group that received the oncolytic virus had very blanched tumors, and they suggested that this is due to the oncolytic virus causing vascular shutdown, which essentially makes the poorly perfused tumor even less well perfused. And I don't know if you can see it on your screens, but this tumor highlighted in the box is very, very light gray pink, whereas the others are a richer red pink color, suggesting poor perfusion. Secondly, as we look at the panel on the bottom right, where they're looking at the 4 treatment groups and semi-quantitatively scoring their metastases, they suggest that the oncolytic virus by itself does not have the ability to reduce the cancer's metastasis, whereas the 3-TSR and the combination group did show reduced metastases. And lastly, they also show that the combination therapies synergistically debulked the primary and secondary lesions. So in the combination group here, the tumor weight was significantly lower than either of the therapies alone. And in terms of metastases, it was also scored significantly lower than either of the treatments alone. Secondly, they performed a microscopy experiment where in each of the panels in the top right, we're looking at vascular endothelial cells in red and vascular parasites in green, with vascular parasites representing representing cells that typically form around mature vessels. And so if we look at the difference between the vehicle-treated and the oncolytic virus-treated cells or mice, the primary tumor had significantly less green signal, suggesting that there was indeed vascular shutdown and loss of this mature vasculature in the oncolytic virus treated group. Whereas in both of the groups that received the 3-TSR anti-angiogenic, you sort of see these somewhat compressed but still ring-like parasite structures. In addition to this, they went on to quantify the hypoxic tumor tissue in a set of images that were not shown in the primary text. But essentially what they show is that the oncolytic virus did not reduce the percentage of hypoxic tumor tissue relative to the vehicle, whereas both of the groups that received the anti-angiogenic did have reduced hypoxia in the primary tumor. Here, sort of a one-off, but they're showing in the primary tumor 3-TSR reduced the intratumoral immunosuppressive cytokine concentrations. And so these are cytokines that might otherwise suppress effector cell function and cause them to go to exhaustion. And so it's somewhat promising that that the anti-angiogenic therapy was able to reduce these the concentration of these factors, and suggests that in combination with oncolytic viruses, which support immune responses via a different route, there could be synergistic action. And these sets of experiments, also assaying the primary tumor, they show that they're looking at the migration of immune cells into the primary tumor. And so here we're looking at macrophages, neutrophils, NK cells, as well as several lymphocytes like the B cells and your CD4 and CD8 T cells. What's indicated in this bar here is that with the oncolytic virus alone, CD8 positive T cells were increasingly trafficked into the primary tumor. But if we look at all the other subpanels with all of the other immune cells, there's no significant effect. And so, yes, the oncolytic virus, the point here is that yes, the oncolytic virus does increase cytotoxic responses to the tumor, but on a broader scale, by itself, it doesn't seem to provoke a broader immune response. Here we look at, highlighted here, we look at the immune cell infiltrates after 3-TSR treatment, where they suggest that 3-TSR treatment increase trafficking of several subsets of immune cells, like your NK and macrophages, as well as your CD4 and CD8 positive T cells. Lastly, though, and perhaps probably the most important point of this paper is that they show the combination of these two therapies uniquely enriches the CD8 T cells, where alone, both of these therapies increase CD8 T cell infiltration relative to baseline. The combination of both of them increased infiltrates by perhaps another fold. And there was also this unique effect of enriching B cells, although I really am not sure as to the mechanisms of how this happened. We mentioned earlier the immunosuppressive cytokines in the primary tumor, such that even if you have infiltrated immune cells, they might not be effective at killing cancer cells because they're suppressed by these immunosuppressive cytokines. And so here they wanted to look at the phenotype of the infiltrated cells to make sure they were actually, able to carry out killing. And so on the panel on the left, they show that the oncolytic virus, both alone and in combination, was able to promote activated NK cell phenotypes, which is great and expected. And that on the right, 3-TSR increased intratumoral proliferating CD8 T cell populations. And so the important distinction here is that they're talking about active NK cells and proliferating CD8 T cells. So proliferating doesn't necessarily mean cytotoxic, but I'll touch on that a little bit later. And it's also interesting that there was no significant difference between the oncolytic group and the vehicle group in terms of proliferating infiltrated CD8 T cells. So the takeaways from these series of experiments was that the oncolytic virus alone did increase the trafficking of CD8 positive T cells, but that the trafficked T cells are not actively proliferating. Again, there's no statistical difference. Even though they look pretty similar, I'm guessing this error bar goes quite far down. And so this is likely due to the oncolytic virus-induced vascular shutdown and hypoxia, with because the tumors are even more poorly perfused. There's probably that buildup of the lactic acid I mentioned on one of the very first background slides that's causing these T cells to infiltrate and then immediately become exhausted because there's no nutrients for them and there's no oxygen. A second key takeaway is that 3-TSR stabilizes vasculature, as we saw on the microscopy subpanels, attenuates hypoxia, and reduces intratumoral levels of these immunosuppressive cytokines. And that even alone, the 3-TSR therapy resulted in broad immune cell subpopulation recruitment. And that the combination therapy resulted in this unique effect of B cell infiltration and CD8 T cell infiltration enhancement. I thought I had another bullet. All right. And so these 2 points are the key takeaways. And lastly, I did want to talk about one of their other figures that's a bit of a one-off again, where they're looking at the interferons produced as a result, or as a result of the viral therapy. And so this Newcastle disease virus that they use as their oncolytic virus has a well-documented sensitivity to interferon beta, which is a type 1 interferon, typically secreted by fibroblasts. And they wanted to capture just how sensitive is this in the context of their study. And so in the panel on the top, what they did was they're using the same 3 cells we mentioned earlier. So IDA is your mouse cancer cell, and CAOV3 is your human cancer, and this is your normal epithelium, the nose. And what they're showing is that the normal cells over 3 days post-infection produce the most interferon, whereas the human cancer cells look like they're producing almost none, and the mouse cancer cells are producing some, but it's significantly impaired, especially by day 3. On the subpanel on the bottom, what they're doing is looking at cell survival after infection and administration of interferon beta. And so they're trying to see if that additional dosing of interferon beta will reduce cancer killing by the oncolytic virus, and if that reduction is dose dependent. And so what they did was they took the ID8 of mouse ovarian cancer cells infected at MLI of 10, and then the top curve they didn't add interferon beta, and on the bottom they did add interferon beta at each of these increasing concentrations. And what they're showing is that the higher titers of interferon beta sort of desensitize the tumors to the oncolytic virus driven death. Is that what's happening? Sorry, this figure doesn't make sense to me. Okay, anyways. The point being, the cancers are shown to be less susceptible to the oncolytic virus if there are high levels of interferon beta. All right. Let's pretend that last slide didn't happen. So in sum, what the authors show is that 3-TSR and the oncolytic virus both individually and synergistically promote trafficking of immune effector cells to the tumor with slightly different subsets that they each recruit and activate. They show that the oncolytic virus recruited CD8 positive T cells are shifted towards a proliferating phenotype. However, as mentioned previously, the increased trafficking and proliferation does not necessarily translate to tumor clearance, and that even if your lymphocytes are infiltrated, immune checkpoints can still derail anti-tumor immunity at this last, most crucial step, and sort of render all the previous buildup pointless. And lastly, that the interferons induce immune checkpoint expression, and they sensitize T cells to exhaustion. And so it would have been nice if the authors checked for effector cell exhaustion markers, like PD1, for example. And so if I take us back to this slide, if they looked at activated CD8 T cells as well as activated NK cells, this might have been a lot stronger, because they show that they would have shown that the infiltrated T cells were actually active and cytotoxic. And then there is evidence within this paper that this might be true, so it's a little puzzling why they didn't show it. On the slide I briefly went back to, they show that 3-TSR increases NK cell trafficking and activity. Hold on. Yeah, they show the virus increases NK cell activity, and they also show that 3-TSR can increase trafficking of NK cells, but that these infiltrated NK cells are not significantly more active. And so it would have been important to elucidate whether the same thing is happening in the CD8 T cells, but they don't, so that's unfortunate. Yep, okay. Lastly, just throwing out a couple things that cross my mind that aren't directly relevant to this paper. So other things that exist are oncolytic viruses with transgenes for interleukins, cyto and chemokines that can recruit immune cells, GM-CSF, and even whole antibodies and bispecific antibodies, which is completely bonkers in my opinion, because what you can do is have this DNA oncolytic virus infect a cancer and then cause the cancer cells to start producing antibodies against immune checkpoints, which is like a 2-in-1. So I'm fairly certain this is an area of active and exciting research. And then other things that might exist that I haven't looked into at all are oncolytic viral therapy for blood cancers and hematological malignancies, which again, keep in mind the difficulty of systemic administration of these OVs might be a little difficult, as well as to the diffuse nature of the cancer might render a lot of the benefits of OVs pointless, because they're not in a highly dense environment, and subsequent infection of cancer cells might be a little harder than if you just inject them intratumorally into a solid tumor. But I believe that is all for today. So if anyone has questions, I will take them, and I will also stop recording. How do I stop recording?